Imagine for a second that you are standing in front of this massive room-sized switchboard.
Oh, boy.
Yeah, and there are over 20,000 individual switches. And your entire job is to figure out what every single one of those switches does to a giant factory. So like you flip one, you run around the factory with a clipboard, you write down every tiny change in the machinery.
Right.
Then you run back, you flip it back, flip the next one, and do it all over again. 20,000 times.
Well, I mean, it sounds completely exhausting and mathematically almost impossible to do efficiently.
Exactly. But that switchboard is the human genome. And flipping those switches one by one to see what happens to a cell, well, that is a very real process in biology. It's called a genome-wide single-cell perturbation screen.
Yeah, which is a mouthful, but it's a huge deal.
It is. The problem, though, is doing this on a large scale currently bankrupts laboratories. But a team of researchers at the New York Genome Center, so this is Alexander Bradu, John Blair, Isabella Grabsky, and Rahul Satija, they've developed a technology called VIPerturb-seq. And they've essentially figured out how to break the million-dollar cost barrier of mapping the entire human genome's functions.
It's massive. It fundamentally changes the economics of modern biology.
Yeah.
Because, right now, the bottleneck isn't the genetic editing itself. We have tools like CRISPR to knock down or mutate genes pretty easily.
Yeah.
The real bottleneck is the reading part, the sequencing.
Okay, so let's unpack this for our deep dive today. Why is combining CRISPR and single-cell sequencing on a massive scale so incredibly expensive right now?
Well, it comes down to a linear scaling problem. I mean, the traditional method out there is known as Perturb-seq. You use CRISPR to knock down a gene, and then you sequence the cell's RNA to see what changed in its behavior.
Right.
But if you want to test all 20,000 genes in the human body, you can't just test one cell per gene. Biology is noisy, like really noisy. You need to sequence hundreds of cells for each individual gene just to get statistically reliable data.
Wow, okay. So we're talking about sequencing millions of individual cells for just a single experiment.
Yeah, exactly. And at conventional laboratory costs, sequencing that many individual cells easily tops $1 million.
Good grief.
Right. It's just not practical for routine science. So researchers usually have to compromise. They either settle for testing a small handful of genes.
Which kind of defeats the purpose of looking at the whole genome.
Exactly. Or they pool all their grant money for years into one massive, slow experiment.
And that is exactly where VIPerturb-seq comes in. Now, the VIP stands for very important perturbations. And from what I understand, it lets scientists act like a bouncer at a club, only letting the absolute most important cells into the expensive sequencing machine. Or if they want to sequence everything, it uses this brilliant chemical workaround to make the whole process dirt cheap.
That is the core of it, yeah. But to really appreciate how they pull this off, we have to look at why researchers are forced into this expensive corner in the first place.
Yeah.
It has to do with how scientists isolate specific traits in cells.
Okay.
It's a process called phenotypic enrichment.
Right, because cells don't just walk up and tell you what they're doing.
Yeah.
If I want to find a cell that is, say, producing a very specific protein linked to a cancer metastasis, I have to go looking for it.
Yeah, and you do that by staining the protein with an antibody that glows.
Makes sense.
But here's the physical problem. The protein you want to study is usually trapped inside the cell. And cells have membranes specifically designed to keep things like large antibodies out.
Right.
So to get the antibody inside, you have to poke microscopic holes in the cell membrane. This is called permeabilizing.
But wait, if I poke holes in a water balloon, it just deflates and everything leaks out. Doesn't the cell just completely fall apart?
It absolutely would, which is why before you poke the holes, you have to fix the cell. You treat it with a harsh chemical, usually formaldehyde.
Okay.
Formaldehyde creates these rigid chemical crosslinks between all the molecules. It essentially freezes the cell in time, locking all the internal proteins in place.
Oh, like bugs trapped in amber?
Yes. Perfect analogy. Once it's fixed like that, you can poke all the holes you want, flush the glowing antibodies inside, and the cell holds its shape perfectly.
Okay, that sounds like a flawless solution. You freeze the cell, poke the holes, stain the protein, and now you can find the exact cell you want. So why is this a bottleneck?
Because the formaldehyde is a really blunt instrument. I mean, it is fantastic at preserving proteins, but it completely shreds RNA. It chops the cell's long, delicate RNA strands into tiny, fragmented, useless pieces.
Oh no, and RNA is what we need for the sequencing part.
Exactly. Standard single-cell RNA sequencing relies on a process called reverse transcription. You introduce an enzyme that physically attaches to one end of an RNA strand and literally walks all the way along it, reading the sequence step by step to create a readable DNA copy.
So if the RNA is chopped up into, like, fragmented confetti, the enzyme tries to walk along the path, hits a break, and just falls off.
Yeah, the reaction fails entirely. You get absolutely no data.
So for you listening out there, think of it this way. It's like having to read a massive, thick book cover to cover just to prove what book it is. But if the pages are ripped and completely out of order, you can't do it.
Right.
So how does VIPerturb-seq get around the fact that the cell's RNA is completely shredded?
Well, they realized that trying to read the whole book was the actual problem. So they abandoned reverse transcription entirely.
Wow, just threw it out.
Yeah. Instead, they adapted a platform called the 10x Flex system. And this uses probe-based detection. So instead of trying to read the whole RNA strand, they engineered two incredibly small chemical probes, a left probe and a right probe.
Okay.
These probes are designed to ignore all the chaos and seek out one specific tiny sequence on the degraded RNA.
Ah, like just scanning the barcode on the back cover of the book?
That's a great way to think about it.
Yeah.
When the left probe and the right probe sit down right next to each other on that tiny surviving RNA fragment, they chemically lock together.
Yeah.
They ligate.
But wait, if the RNA is shredded into 1,000 pieces, aren't you worried that the specific tiny barcode section you're looking for also got chopped in half?
I mean, it does happen. But because the sequence the probes are looking for is so incredibly short, the mathematical probability of finding an intact snippet is vastly higher than the probability of finding a whole, pristine RNA strand.
Right.
You don't need perfect RNA anymore. You just need a tiny landing pad.
So the logic here is incredibly clean. Fixing cells to find specific proteins destroys the long RNA strands needed for traditional sequencing. But by switching to these lock-and-key probes, you bypass the need for pristine RNA entirely. You can freeze the cell, stain it, shred the RNA, and still get perfect data.
Which completely opens up what we can study. I mean, if you could pause a cell right in the middle of a disease state like a neuron, the exact moment it starts forming an Alzheimer's plaque, you could freeze it, probe it, and read its genetic state perfectly.
That is wild. So before we move on, let's do a quick recap for everyone listening. The million-dollar problem is sequencing costs. To find specific cells, we have to fix them, which shreds RNA. But VIPerturb-seq fixes this by using left and right probes that just scan a tiny, intact barcode, skipping the need for whole RNA.
Exactly.
So here's a question for you to chew on as you listen. If you had this tool, and you could pause a cell right in the middle of any disease state like a cancer cell metastasizing, what specific protein would you want to freeze and study? Because this tech makes that possible. But it also introduces a massive, terrifying math problem, doesn't it?
Oh, yeah. The cost of the probes.
Right, because the probes solve the degraded RNA problem for the cell's natural genes. But in this experiment, you are using CRISPR to aggressively mutate the genome. And to hit every single gene, this New York Genome Center team used a massive library of over 57,000 different CRISPR guide RNAs.
Right. And if you have to design and purchase a custom left and right probe for all 57,000 of those guides, the cost becomes astronomical. You've basically solved the sequencing bottleneck, but created a chemical manufacturing bottleneck.
Yeah, that doesn't really help the budget. So how do you track 57,000 different CRISPR mutations without buying 57,000 unique probes?
This is where they introduced the split-barcode system. Instead of trying to capture the actual CRISPR guide RNA sequence directly with a probe, VIPerturb-seq attaches a synthetic dual-part barcode to each guide RNA.
You are essentially tagging the tag.
Exactly.
Okay, but doing this inside a living cell has to be tricky. I know researchers use lentiviruses to deliver these CRISPR guides into the cell. They basically hollow out a virus and use it as a microscopic delivery truck. But are viruses reliable enough to handle these dual tags?
Well, they are actually notoriously sloppy during the packaging phase. Viruses undergo a process called recombination. So lentiviruses actually have two copies of their RNA genome, and when they are packing their genetic material, the enzyme that copies them frequently jumps back and forth. They mix and match different pieces.
Oh, I see. So you could design a beautiful experiment where barcode A is securely attached to a cancer gene mutation. But during delivery, the virus fumbles it, swaps the tags around, and suddenly barcode A is attached to a mutation for eye color.
Which would completely ruin the data. You'd be drawing heavy conclusions about cancer based on an eye color gene.
Yikes. So how did they stop the virus from swapping the tags?
To prevent this, the researchers had to force the virus to keep the guide and the barcode together. And they did this through extreme physical proximity. They place the synthetic barcode less than 100 base pairs away from the guide RNA.
Just literally gluing them so close together that the virus's clumsy machinery can't physically wedge between them to swap them?
Exactly. They become an inseparable package deal.
Okay, so the tag is secure, no swapping. But we still haven't solved the cost issue. How does a dual-part barcode stop them from having to buy 57,000 unique probes?
Well, it's pure combinatorial math. They designed a reusable library they call the guest list. Instead of one long barcode, the tag is split into a left-hand side and a right-hand side. They only designed 238 left halves and 240 right halves.
Oh, I see the math. 238 multiplied by 240, that equals over 57,000 unique combinations.
57,120 to be exact. So instead of buying 57,000 custom probes, they only need to purchase 478 probes.
Wow.
Yeah, and by mixing and matching how those 478 probes combine, they can uniquely identify every single genetic perturbation in the entire human genome.
It's exactly like a first name and a last name. So for you listening, think about it like this. You only need to memorize a few hundred common first names like John or Sarah, and a few hundred common last names like Smith or Lee.
Right.
And by just combining them, you can uniquely identify tens of thousands of different people in a city. You don't need 57,000 completely unique single names.
That is a perfect way to visualize it. But they couldn't just pick random sequences of DNA to act as these names. DNA is sticky and unpredictable, so they imposed incredibly strict biochemical rules to make sure this guest list library was flawless.
Right, because if your first name and last name tags get stuck together wrong, you're back to ruined data. What kind of rules did they use?
Well, first, they instituted a strict Hamming distance threshold.
Okay, I've heard that term in computer science. It's an error correction concept, right?
Yes, exactly. It means every single barcode half must be chemically distinct from all the others by a minimum number of letters. Think of it as a built-in spell checker or a minimum edit distance.
Right.
Sequencing machines aren't perfect. Occasionally they misread a letter of DNA. If there's a one-letter typo during sequencing, the Hamming distance ensures that the typo doesn't accidentally turn the barcode for Smith into a valid barcode for Smith.
So a tiny machine error doesn't accidentally ruin the cell's identity. If the machine reads a typo, it just knows it's an error rather than misidentifying the gene entirely.
Exactly.
Yeah.
You'd need the machine to make 3 or 4 specific typos in a row to accidentally hit another real barcode.
Which is statistically almost impossible.
Right. And second, they tightly balance the GC content. In DNA, guanine and cytosine form stronger physical bonds than adenine and thymine. If a synthetic barcode has too much GC, it sticks too tightly to things it shouldn't.
And if it has too little.
It doesn't stick enough and just falls off. They also had to use structural algorithms to ensure the barcodes wouldn't accidentally fold in on themselves.
Like a piece of tape folding over and sticking to itself before you can actually use it.
Yeah, forming a 3D hairpin structure. That would physically block the probes from landing. And finally, they took every single synthetic barcode and cross-referenced it against the entire human and mouse genomes to ensure there was absolutely zero accidental matching with natural RNA.
So what they built is a highly engineered, typo-proof, biologically invisible master key, a reusable library of just a few hundred tags that combine to track tens of thousands of mutations. And you can just pull this off the shelf for any future experiment.
Which is exactly what makes VIPerturb-seq so scalable. The chemistry is solved, the barcodes are cheap, the degraded RNA is readable.
So time for another quick recap. To avoid buying 57,000 probes, they split the tags into a first name and last name system called guest list. They keep the tags close to the guide RNA so the virus doesn't fumble them. And they made the tags typo-proof and structurally perfect.
Spot on.
So for you listening, think about the sheer efficiency of combinations here. How else could this first name, last name tagging system be used in other fields of science or data to track massive amounts of information cheaply? It's a really powerful concept. But that leads us to the next massive hurdle. We still have to physically process hundreds of thousands of cells through a sequencing machine, right?
Yeah.
Because earlier we said the machine time itself for millions of cells costs a fortune. Did they find a way to make the physical sequencing cheaper too?
They did, by superloading the machines. The researchers tested VIPerturb-seq in 2 different ways. The first was an unbiased screen where they literally test every gene at massive scale to see what happens. To do this affordably, they utilized a technique from the 10x Genomics Flex V2 platform called combinatorial indexing.
Okay, let's break that down. How does combinatorial indexing stop the sequencing machine from basically choking on 500,000 cells?
Well, in standard single-cell sequencing, you push cells through a microfluidic machine that encapsulates each individual cell into a tiny fluid droplet.
Right?
And inside that droplet, the cell's RNA is tagged with a droplet barcode, so the computer knows which cell the RNA came from. The problem is, the droplets are the expensive part. If you cram too many cells in at once to save money, 2 cells end up in the same droplet, they get the exact same barcode, and your data is ruined.
Oh, because the computer thinks it's looking at one cell, but it's actually looking at a mashed-up Frankenstein of 2 different cells.
Exactly. So normally you have to run them through slowly and use an enormous amount of expensive droplets.
Unless you add an indexing step before the droplets.
Right. With combinatorial indexing, they physically split the cells into 24 separate plastic wells first. In each well, they add a unique chemical well barcode to the cells. Once every cell has a well barcode, they pool all the cells back together, mix them up, and then run them through the microfluidic machine to get their droplet barcode.
I see. It's like adding a zip code to a letter before you sort it into individual mailboxes.
Exactly. Even if 2 letters end up in the same exact mailbox as same droplet, it doesn't matter anymore.
Because the computer can look at them and say, they share a droplet barcode, but this one has the well barcode from well 3, and this one has the well barcode from well 12. They are clearly distinct cells.
Because of that dual labeling, the post office, the sequencing machine, can process massively more mail in the exact same facility without mixing anything up. In a single lane of the sequencer, they profiled an astounding 440,000 cells.
That is a staggering amount of data for one lane.
It's a 50-fold increase over traditional methods. And they didn't sacrifice sensitivity either. They recovered a highly detailed readout of the gene expression, about 15,100 unique RNA molecules per cell.
Okay, so the mechanics work flawlessly. The throughput is huge. But let's talk about the biology. When you just unbiasedly blast the whole genome and read 500,000 cells, does it actually reveal anything new?
It does. The sheer volume of data allows you to find entirely hidden relationships. For example, in this unbiased screen, they looked at the results of knocking down a gene called UVSSA.
UVSSA, what does that normally do?
It's traditionally known to be involved in transcription-coupled DNA repair. Basically, it fixes physical damage to your DNA.
Okay.
But when they looked at the massive data signature from the VIPerturb-seq screen, the downstream effects of knocking down UVSSA perfectly mirrored a completely different biological process called nonsense-mediated decay, or NMD.
I've read about NMD. That's the cell's internal spell checker, right? The system that hunts down and destroys garbage, mutated RNA before it can be translated into broken, harmful proteins.
Precisely. They found that knocking down the DNA repair gene changed the behavior of known RNA spell-checking targets, like a transcription factor called ATF4.
Wait, why is that a big deal? They're both just maintenance systems, right?
Well, think of it this way. It's like assuming the plumbers and the electricians in a skyscraper work completely independently. And then suddenly, by looking at massive amounts of data, you discover they are communicating on a secret shared radio channel to manage the building's power.
Oh, wow.
Right. It proved that UVSSA isn't just fixing DNA. It has an intricate hidden relationship with the RNA spell-checking system. It shows the incredible power of unbiased screening to connect dots we didn't even know were on the same page.
So to recap this section, to scale up to 500,000 cells, they use combinatorial indexing. They add a well barcode before the droplet barcode, like a zip code. This gave a 50-fold increase in cells processed. And biologically, they proved it works by finding a hidden link between a DNA repair gene and the cell's RNA spell checker.
That's the unbiased approach in a nutshell.
So for you listening, think about this. When you have the power to map hundreds of thousands of cells cheaply and find unexpected connections, what complex biological mystery like aging or how memory formation physically alters neurons do you think this kind of unbiased mapping should tackle next?
It's an exciting thought.
It is. But I want to pivot because scaling up to 500,000 cells is great if you want to look at everything. But what if I don't care about the whole genome? What if I am a researcher who is obsessed with finding the exact genes that control just one specific physical trait?
That brings us to the true power of the VIP bouncer approach. And this relies heavily on that phenotypic enrichment, the glowing antibodies that we talked about at the very beginning.
Right, the very important perturbations. How do they actually bounce the cells that aren't important?
They use fluorescence-activated cell sorting, or FACS. Remember those fixed, permeabilized cells with the glowing antibodies inside?
Yep, the bugs in amber.
You run those cells, single file, past a highly sensitive laser. The laser measures exactly how bright each individual cell is glowing. And the machine physically sorts the cells into different tubes based on that brightness.
Literally acting like a bouncer at a club looking at VIP wristbands. If your wristband isn't glowing bright enough, you get kicked out of the line. You don't get into the sequencing tube.
Exactly. And to prove how powerful this bouncer is, the researchers did a case study on a protein called vimentin.
Why vimentin?
Vimentin is an intermediate filament protein. You can think of it as the cell's internal scaffolding, kind of like the rebar in concrete.
Okay.
It controls how squishy or stiff a cell is, its mechanical structure, and how it responds to physical stress. If vimentin levels are dysregulated, it can severely disrupt cell division and signaling.
So it's super important for the physical structure of the cell.
Very. The researchers wanted to find out exactly which genes in the entire genome act as negative regulators of vimentin, meaning if you knock the gene out with CRISPR, vimentin levels shoot way up.
So they took a batch of cells, I think they used K562 cells for this, applied the entire genome-wide CRISPR mutation library, let the cells grow, and then stained them with a glowing antibody specifically for vimentin.
Exactly. And then they brought in the VIP bouncer, they used the laser to look at all the cells, and they threw away 97% of them.
Wow, 97% just in the trash.
Yep, but they kept only the top 3% of cells that were glowing the absolute brightest for vimentin.
That is absurdly efficient. By throwing away 97% of the cells, they didn't have to sequence millions of cells to find the needle in the haystack. They just threw away the hay and kept the needles.
Exactly. Because of that enrichment step, they only had to sequence 12,000 cells to get a definitive genome-wide result, 12,000 instead of millions.
And what did those 12,000 super bright cells tell them?
Well, they looked at which CRISPR barcodes were inside those 12,000 cells, and the screen overwhelmingly pointed to a group of genes that make up something called the regulator complex, along with FLCN. So genes like LAMTOR1 through 5, RIGA, RIGC.
Wait, is that surprising?
It was a massive surprise. The regulator complex is famous in biology textbooks for doing something entirely different. It sits on the surface of the lysosome, which is basically the cell's stomach, and it acts as an amino acid sensor.
Okay, so it monitors food.
Right. It monitors whether there are enough amino acids available. If there is food, it turns on a massive growth pathway called mTORC1.
So it's a food sensor, but the VIPerturb-seq screen is saying it also controls the physical scaffolding of the cell.
Yes. It proved that this complex doesn't just sense food. It actively regulates the abundance of vimentin.
But are we sure about that? Because biology is messy. Couldn't the fragmented RNA just be giving a weird, noisy signal?
The researchers actually anticipated that skepticism. Because the Flex system uses probes, it can capture both RNA tags and protein-level antibody tags at the exact same time.
Oh, nice.
Yeah, so they cross-validated the discovery using independent protein data. They proved definitively that knocking down the regulator complex physically caused vimentin protein levels to spike.
That is incredible. Food sensing and structural squishiness are physically linked by the exact same complex. That is a completely elegant discovery.
It really is.
So, final recap. The VIP bouncer uses a laser to sort cells by brightness. They tossed 97% of the cells, sequenced just 12,000, and discovered a brand new role for the regulator complex in controlling cell squishiness.
Perfect summary.
And it makes you wonder about the flip scenario. We went from millions of cells down to 12,000 just by looking at the top 3% brightest VIPs. But what if we looked at the bottom 3%, the cells that utterly fail to glow? Could that be how we isolate the exact mutation that makes a cancer cell entirely immune to a chemotherapy drug?
That is exactly the kind of targeted question this platform enables.
Yeah.
I mean, when we zoom out and look at the bigger picture, VIPerturb-seq fundamentally shifts the economics of discovery. By abandoning reverse transcription for lock-and-key probes, by utilizing the split-barcode math to make tagging cheap, and by allowing for either targeted VIP bouncing or massive combinatorial indexing.
It's doing it all.
Yeah, this technology turns a $1 million pipe dream into a routine afternoon laboratory experiment.
And it's fully generalizable, right? You don't just have to use CRISPR to knock genes down. You could swap out the CRISPR guides for tools like ORFs that force genes to overexpress, or tools that test entirely synthetic genes. It is basically a universal remote for that massive 20,000 switchboard we talked about at the beginning.
Absolutely. It essentially removes the friction of gathering the data.
Which leaves us with a fascinating, slightly intimidating new reality. Because if generating the data is no longer the bottleneck, the new bottleneck becomes interpreting it.
Oh, for sure.
So for everyone listening, I want to leave you with this final thought. Once we use VIPerturb-seq to map out a perfect 100% complete virtual cell on a computer, a simulation where we know exactly what every single genetic switch does in every possible scenario, will we finally be able to test and perfect new drugs entirely in software before they ever touch a human patient? Take a deep dive into that thought.